on language and hemispheric specialization, but before I get started, what I wanted to do is just go over one really key point from the last lecture. And this point was something that was kind of new to the categories of things that we've been talking about. Remember, we talked about, the lecture last week was focused on the executive functions of the frontal lobe, what the frontal lobe does. And like in many other of these higher cognitive functions, we learned a lot about this by lesions. So you had Phineas Gage with this huge frontal lesion. We had the example of animals with very selective lesions that would cause an impairment on this delayed response task that you see at the top here. So I forgot my pointer. Okay. So the delayed response task is a very selective lesion. This slide, this part of the slide down here just shows you the small part of the frontal lobe. When just a small part was damaged on both sides, you get a severe impairment of the animal's ability to perform this task. And we went over possible ways, reasons why the animal would perform poorly. He can't see the things. He can't reach for the things. You can control for that. Yes, he can see. He can discriminate between things. He has perfectly good motor function. But he can't seem to remember and keep in mind the location of this favorite little morsel of food during this very short delay interval. And what we came to understand is that what this damage to this area is impairment in what we call working memory, memory for keeping things in mind. We talked about all the different situations in our everyday life that we use working memory. Now, one very important aspect that I mentioned but I want to emphasize is that the beauty of this task is that there's a lot of interference. For all the trials, the thing to be remembered is either the right side, left side, right side, left side. So in a sense, the animal has seen the right side and the left side baited an equal number of times. He can't say, okay, what was the most overall, which side is the most familiar or not. He really has to remember, okay, on this trial, I have to remember right side, right side, right side. That working memory component is very critical. Now, I also want to contrast this task to another task, key task that we've talked about before, the delayed non-matching to sample task. I told you this was a task that taps long-term memory. And one of the key differences between this delayed response task and this delayed non-matching to sample task is that there's not a lot of interference. Because each of these items used on the delayed non-matching to sample task are shown only once during the trial. So they only see this coffee kettle once, and they only see the fire engine once during the test, and on the next trial there's new objects. So what the animals mainly do here, and what they could take advantage of, is their recognition for the familiarity of these different items. That is a key function of the medial temporal lobe. And as you make the delay between the sample and the test longer, that is what really taps medial temporal lobe function. So the fact that these are unique objects makes this task particularly sensitive to medial temporal lobe lesions, and it's also quite sensitive even it's the most sensitive, I should say, at the longest delay intervals. This contrasts with the delayed response task, where there's lots and lots of interference. Animals cannot use relative familiarity of these two different boxes here to be able to solve the task. They have to use their working memory. Okay? So that's one of the points that I wanted to emphasize. The second point is that in lots of different situations, in lots of different studies that we've talked about, lesions have played a really important role in allowing us to understand the higher cognitive functions of the brain. So we started with this delayed non-matching to sample task, which this is a task used in monkeys. We talked about the study by Mort Mishkin who used this task to show, demonstrate for the first time that monkeys with lesions similar to HM did have a devastating memory impairment, particularly at the longest delay intervals. Similarly, we have this very selective lesion in the frontal lobe causing devastating deficits on the delayed non-matching to sample task. But for the lecture last time, we went one step further. Not only do we learn about the functions of a brain area through a lesion, but we then said, okay, if this task is so sensitive, what are neurons doing? What is the pattern of activity, the pattern of spiking that neurons in this area are giving us as the animal is performing this task? That gets even deeper into what is the code that these neurons in this key area that we identify are using for this critical cognitive function, working memory. Okay? This is the first time we've really gotten into this level. The only other time that we talked about something similar is habituation and long-term potentiation. But this was the first time we really related it to exactly what the cells are doing during a task. So let me just, with that introduction, let me just remind you what we talked about. The task that they used, this what's called behavioral neurophysiological approach. So behavioral neurophysiology, because the animals are behaving, and you're doing neurophysiology at the same time you're recording the activity of brain areas as animals are performing the task. It's actually a very, very powerful tool, and I'm giving you just one example. The task that's used, to remind you, is the oculomotor delayed response task, ODR. And first, animals fixate on a central fixation spot. The animals are cued. They can't look at this cue. They have to maintain fixation. After delay, without the cue, they have to make a saccade to the remembered location. So during this time, this is the working memory time. And behaviorally, these are behavioral results here. You can see the dots indicate the final saccade location of where the eye ended up. And here's the locations when the dots are there. So he has a target to go to, so he's very precise. And without the targets in the actual task, you see he does a little worse, but still okay. And after a longer delay, 6 seconds delay, he does a little bit worse, but still okay. That's just behavior. What are the cells doing during this task? We're particularly interested in the delay interval. That's where memory for this location has to be kept in mind. And remember, here is what we got. Each one of these graphs show you the response of the same cell that I'm recording as the animal is performing this task. And so here are trials in which the target was up here, 135 degrees, lots of different trials here. And you can see during the fixation period, the cue period, the delay period, and the response period, the cell is basically not changing its firing rate. You can look at individual trials here, and this is the average response of all these trials added up down here. Here, you can see a little bit of a response during the motion part of the task. This is when the animal makes this isocotic response. But of course, the key trials are the ones down here at 370 degrees, the downward movement. You can see that the cell really fires. It starts to fire a little bit during the cue, but maintains its firing rate during this delay interval. Remember, there's nothing on the screen. This is a time when the animal just has to maintain this information in mind, just like you're retaining a telephone number in mind. So what this tells us is individual cells in this dorsolateral prefrontal cortex are encoding this working memory, this keeping in mind memory, also called scratch pad memory, by maintaining their activity during the delay, but in a selective way, only for particular directions that the animal has to remember and not for other directions. So this cell is very selective for remembering the bottom target down here, the south target. You can imagine other cells, and in fact they showed that other cells in other areas of the dorsolateral prefrontal cortex might be sensitive to this direction, the rightward direction, or other cells to the leftward direction. So all these cells together are helping us keep things in mind for all possible directions that the animal has to remember. Okay? And so what I also mentioned is that this individual cell, the activity of this individual cell in all these different directions looks like a tuning curve, but this is a memory tuning curve. A tuning curve for the memory of particular things. Okay? So this is yet another deeper question that we can ask about how these different areas are contributing to different cognitive functions. You can ask the same thing, we didn't talk about it, but how are cells responding? What if you record as animals are performing a delayed non-matching to sample task? We didn't talk about that. So for this task, where would you like to record in the brain? What would be a reasonable area to record in as an animal is performing a delayed non-matching to sample task? What do you think? What would be a reasonable area?
During the delay interval?
Not what time interval, but where in the brain would be a good area to record, given this approach. The medial temporal lobe, thank you. Perfect. So you record in the medial temporal lobe because that is the area which, when damaged, causes impairment on this task. That tells us that the medial temporal lobe is contributing to the performance of this task. And now I'm going to swoop in with this new approach, behavioral neurophysiology, record in a normal animal in its normal brain in the medial temporal lobe to ask not what happens when you damage it, but what are these cells doing in the normal situation as the animal has to remember these stimuli. Now we didn't go over this, but just to give you a flavor, this definitely has been done. And one of the patterns that you see is when a stimulus has been seen before. So this stimulus is a repeat of this stimulus. When you've seen it before, the response to that stimulus actually diminishes. So in the medial temporal lobe loves novelty. It loves to respond the first time you see something new. So my medial temporal lobe was going crazy the first day of class because I'd never seen you all before. You were very, very unfamiliar. Now, my medial temporal lobe is shut down because I know you all, nothing's happening there. But something new happens in class. Something new happens in my experience. My medial temporal lobe really picks that up, and the first time things are presented, it seems to respond more. Okay? Now, I'm not going to ask you about that, but that's just to give you a flavor for how we use this kind of tool. Any questions about prefrontal cortex differences between delayed response and delayed non-matching to sample? This is a very, very important distinction, and this is definitely going to be on the test. So any questions about that? Okay, great. So let's start with the topic of our lecture today, which is language. So language is one of the things that makes us unique in the animal world. Animals can communicate with each other. Whales have whale calls, birds have bird song. But the attachment of meaning to these signals is quite unique in humans, and I just show you some examples of authors that were able to use language in a particularly meaningful and deep way. So it's not just an area, a tool for communication, but it can be used as an art form as well. So I want to start a little bit with development. So we only have one lecture on language, so there's so much to understand. But we need to start with the beginning. What are the basic elements of language? I want you to learn these terms. Phonemes are basic speech sounds. Just all the basic sounds, and different languages have different phonemes in their vocabulary. So Basque language, which is very, very unusual, and those words tend to start with X and Y's, much more than you would think possible, has a very different phenome than English. Morphemes are simple units of meaning. Semantics, as you probably all know, is meanings of words or sentences. So I always find it fascinating that from birth, babies are babbling. Everybody knows that babies babble. And as we analyze the babblings of babies, we have learned that the sounds that they make in these babblings have elements from phonemes from all languages. So it's as if we're born with this ability to actually make these baby sounds, these babblings, but that have the possibility of morphing into all possible phonemes known. There are thousands of identified languages, and only a subset of them have been clearly and carefully studied. So that means that any baby can learn any language. And what it depends on is experience from hearing spoken language, from mom speaking to them. There's something in language research called motherese, that all mothers in all cultures speak to babies in a certain way. We all tend to do it. You speak baby talk to babies, but that kind of lilting way of speaking emphasizes both meaning and sounds of the particular language that you're speaking. So what you're doing to the baby, what you're doing with the baby as you are speaking your version of motherese to the baby, is you're focusing the baby on those particular sounds from your language. So, and that just brings up another very important concept, which is the concept of critical periods. I think we might have talked very, very briefly about critical periods in the sensory systems. If you don't have vision, a particular kind of visual stimulation at an early age, your vision will not develop normally. And we know from studies in which children, either by accident or because of terrible circumstances, were brought up without any auditory input, no interaction with other people. And in that situation, if you do not get verbal interaction and language training through this kind of motherese talking, you will never learn how to use language properly. There's a critical period of language. There's a critical period of language learning. Perhaps all of you, some of you can appreciate that it was much easier to learn languages when you were younger. And as you get older, in your old age now as college students, it might be a little bit harder to learn. Has anybody had that experience where it's much easier to learn things earlier than later? Okay, so that is what you're experiencing, is the critical period for language. You guys actually, despite the fact that you're very young, you're kind of reaching the end point of your critical period, and it's much better to learn it when you're younger as a newborn. Learning multiple languages, that can come much easier than if you wait longer and longer and have not and don't develop that ear for the different phonemes that you do so naturally when you're a baby. Okay, so just like in declarative memory function, just like in fear memory function of the amygdala, just like in executive memory function, the study of language has benefited from brain damage and patients with brain damage. Aphasia is a collective deficit in language comprehension and production. So these brain areas that we're going to talk about have dual capacities. Some of them help with the comprehension of just auditory information coming in, and the other parts of these language areas are helping with the verbalization, the speaking of language. A scary fact is that 40% of all strokes produce some aphasia, so some language impairment, and you may have had a direct experience with that with friends or family members that have had strokes and have either language production problems, they have problems speaking, or more difficulty understanding. So there are two historical names that I want you to know about in terms of language production and understanding. The first is Paul Broca. So Broca was a doctor in France in the mid part of the 1800s. And a patient came to him with a leg infection. And at the time that Broca treated this patient could only say one thing, tan. So he would try and answer everything with this word tan. Tan, 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 tan. Didn't make any sense. the person could understand what we were asking but couldn't produce any sound other than tan. Now, unfortunately, the patient, right after he came to Dr. Broca with a leg infection, happened to die just a few days later, and Broca got access to his brain, and what he found was a very, very specific brain lesion in the left inferior frontal gyrus. This is one very important point that, that you should know, that language is lateralized on the brain, in the brain. There's only one side of your brain where these key language areas are, and this first came to light because of this patient had a lesion in the left inferior frontal gyrus and had word production striking impairments. Well, again, kind of like any single case study, you don't know what's going on. It could be other parts of the brain that they couldn't see, they couldn't study in a detailed way. But the very next patient that Broca studied after this patient Tan was another patient. He was an elderly patient. He was about 83 years old. And before he saw Dr. Broca, he had collapsed. And when he came out of this fit, he only had 4 words that he could say. These patients were in France. So there was only 4 words in his vocabulary. There were oui, no, toujours, and trois. Oui and no are yes and no. Toujours is always, and trois is 3. So after being a normal patient, he had this fit. He could only say 4 words. He had some sort of another medical condition, came to Broca, and soon after again died again. And Broca happened to be able, not happened to, but he was very interested in getting the brain after the patient died. And it turns out that the second patient also had a quite selective lesion in the left inferior frontal gyrus. Again, very limited vocabulary, could only utter 4 different words, tried to answer all questions with those words. Here's a picture in France at the Sorbonne Institute of patient Tan's brain. It has been preserved. Obviously, it has not been dissected at all. It's still whole, and you can see the location. This is the left hemisphere. Here's the frontal lobe. Here is the occipital lobe. Here is the lateral sulcus, and just dorsal to the lateral sulcus, relatively anteriorly, is the left inferior frontal gyrus, where Tan's lesion was. Here is a dry picture of the same brain, much higher quality here, so you can see the nature of this lesion. So from these 2 striking patients, Broca identified an area that seemed to be important for spoken word, for word and language production, and he named it, of course, Broca's area. It was his area. Then, a few years later, still in the 1800s, another neurologist, a German neurologist by the name of Karl Wernicke, described a brain area in the same way. A patient came to him. He had problems with language, and the patient died, and Wernicke was able to identify the brain area and described a brain area posterior, but kind of nearby to the area identified by Broca, as critical for language comprehension. So this patient that Wernicke studied with a more posterior lesion, he could speak just fine, but what he spoke was gibberish. So he could string words together. The words were mainly real words. Sometimes they were made-up words, but they made no sense. And so this seemed to be a separate area for language comprehension. So based on these first initial studies in the mid-part of the 1800s, we've come up with 3 main types of aphasia that we know exists, again, from this common effect of aphasias developing from strokes. The first is called non-fluent aphasia, or Broca's aphasia. Difficulty with language production, but fairly good comprehension. The second is fluent aphasia, Wernicke's aphasia. Good language production, you could actually string words together in a regular way, but language does not make sense, and there's very poor comprehension. comprehension. The third category is a combination of the first two, which is global aphasia, total loss of ability to understand or produce language. Given these serious concerns and the effect that it would have on your everyday existence, if you could no longer speak words or you could no longer understand words, even worse, there are very, very specific tests that have been developed if you have to have a brain operation that will involve either the right side or the left side. Most people have their language located in the left hemisphere. However, a small proportion of left-handers have their language area on the right side. You want to know which side of the brain is critical for language before you go in there, because you do not want to be messing with these language areas. And the test for this, which is described very nicely in your book, is called the WADA test. It was developed quite a long time ago, which is a very, very simple, quite crude test they used starting, I think, in the 50s or 60s, where they simply inject an anesthetic into the carotid artery of one side. You have a left and a right carotid artery. Anesthetics that go into the right carotid artery will anesthetize basically your right hemisphere temporarily for a minute, okay? And conversely, anesthetic in your left hemisphere will anesthetize the left, in your left carotid artery will anesthetize the left hemisphere for a few minutes. So you can put this anesthetic in and look at the effect on language production. So what typically happens is you're asking the patient questions, count backwards from 10, tell me your name, tell me where you grew up. And at some point during this questioning, they put the anesthetic typically in the left hemisphere, and they ask whether the patient can still speak. Usually what happens is they cannot. They try. They know that you're trying to, they usually know that you're trying to ask them something, but all verbal communication stops for a few minutes, and then it starts back up again as if nothing happened. And so that is, if that anesthetic has been injected in the left side, we know for sure that the left side is dominant for language. Okay, so where exactly are these brain areas on a prototypical, beautiful human brain that we see here? Here is Broca's areas. Note its position relative to the primary motor cortex. So the actual motor areas for your mouth and tongue are right here. These are more like premotor areas, telling the motor areas what to do, what to say. And this Broca area is right in front of the primary motor cortex. Primary auditory cortex is here, also critical for language. Obviously, you can't understand language if you can't hear it. Wernicke's area is, now we jump down onto the temporal lobe, a little bit posterior to primary auditory cortex in this area. And finally, we're going to be talking about the angular gyrus that gives meaning, that helps give meaning or analyze visual words seen on the page. So all the work that you're doing, reading and comprehending, has a lot to do with angular gyrus. Yes, question? That all depends on the lesion. Sometimes they can write. So these aphasias really only affect spoken and comprehension of language. So of course, if you can't comprehend language, you really can't write anymore. But yes, if you have a more pure impairment of Broca's area, you can still write, because all of that machinery should be completely conserved in you, your understanding and your writing areas in the motor cortex. Any other questions? Okay, good. So, let me just show you PET images of, here is a non-fluent Broca's aphasia. So here we're looking at slices like this, where this is anterior and this is posterior. This is kind of a horizontal-like slice, but they've taken it at slightly an angled position here. So you can see the darker area in this anterior inferior gyrus. Inferior frontal gyrus, I should say, is causing a non-fluent Broca's aphasia. And here are more schematic diagrams using the same format of these are where the slices, semi-horizontal slices are taken, and you see the non-fluent Broca's aphasia. This is anterior, this is posterior in my horizontal slice, and the Broca's aphasia are more anterior. The Wernicke's aphasia tends to be more posterior. The global aphasia is huge. including both of these sides. The other thing I want you to appreciate here is that these lesions are a mess. Okay, these lesions were not made to cause language deficits. They are just lesions that occurred because of strokes. Strokes are very messy. So it's not that easy. You never get a pure Broca's lesion of Broca's area because strokes just don't work that way. And that's very important to keep in mind as you analyze the effects of strokes in patients. Some may be quite striking, yet they're always messy. So you have to keep that kind of caveat in mind. So let me just quickly go to a quick demo just so you can see what a typical aphasic looks like. And we're going to start with the Wernicke's aphasia. So what you're looking for is fluent speech, but not comprehensible speech. And unfortunately, this is the best example I could find. Both the questioner and the patient have quite thick accents, so don't let that fool you. But let's look quickly at the Wernicke's aphasia, and then we'll quickly look at Broca's aphasia. Okay, Wernicke's aphasia. Remember, comprehension is not there, but fluidity is there.
Okay, Bruce, how are you? I am fine. All of dying was fine. Okay, Bruce, okay. Now I want you to do something for me. Can you please explain what you see in the picture right here? Okay, there is a green colored tree, which from the color of the green that can be understood. From the color of the green, that is a tree. Right part of the painting, there's a blue bank from the color of the bank.
Okay, so he understands a little bit. How are you? I'm okay. What's there? I actually can't see whether there is a tree there, but I'm assuming there's a tree. But you can see that he's stringing words together. They don't really make sense. This is a classic case of Wernicke's aphasia. So fluid speech, he's speaking a lot, he's using words a lot, but they're not making a lot of sense. So I'm going to jump forward here. Then we're going to go into Broca's aphasia, which will look and sound very different.
Fred, I got you some questions. What brought you back to this hospital again? Yes, Wednesday, that and mom from the hospital and well at 9 o'clock and then o'clock and then the girl
Okay, hopefully you could appreciate that he has some words, but they're very fewer words, and he has a much harder time getting them out. So one would have to do a deeper analysis of who has better comprehension, this Broca's aphasic or the Wernicke's aphasic. But in general, the Wernicke's aphasic has much worse language comprehension. And sometimes it's hard to tell from these things, just from these videos. But the Broca's aphasic, while he can't get the word out and you can't tell that he understands, there are other tests, for example, written tests that you can do to show that he has better comprehension in general than the Wernicke's aphasic. Okay, so let's go back to, okay. So I want to go over one model. So now we have these two different areas. Broca's area, so you can only, you can either say tan or 4 words if you have this damage to this area, or Wernicke's aphasia, a problem with comprehension, but you babble just fine. The model that I want to go over is a model initiated by Wernicke himself and expanded by Geschwin, and it's the Wernicke-Geschwin model of aphasia. This is also in your book. So, So Wernicke proposed a very simple model, a connectionist model. So the comprehension area is simply connected to the speech area. So language is comprehended in Wernicke's area. Then there is a big fiber bundle we know from the anatomical analysis of these areas from Wernicke's areas to Broca's area. It's called the arcuate fasciculus. This is in your book, and you are responsible for this. And through that connection, the meaning gets translated to Broca's area, where motor preparation for the ability to repeat this information or speak about it is prepared, and then Broca's area has very strong connections to the primary motor areas involved in speech, the tongue and the mouth and all those areas. And that simple idea was that deficits in language represent breaks in a network of these components. And each component is involved with a particular feature. So let's go over, sorry, that's the first part, Wernicke. Geschwin refers to, was focused on the idea of a disconnection theory. This part of the model emphasizes language impairment following loss of connections among brain areas, and each component, kind of like the first part that I talked about here, each component is involved with a particular feature of language. So let's see exactly what they mean. So again, this is the Wernicke-Geschwin model, where different brain areas and different connections have particular roles in our language production and comprehension. So this is speaking a heard word. So you hear a word, what's happening in the brain, what areas are involved as you speak a heard word. So first you have to hear it. This is not surprising. You will be able to tell us, for example, exactly how auditory information reaches primary auditory cortex, okay? But we're going to bypass all of that for now. We're going to assume that you know the pathway, and that primary auditory cortex is where this information is processed. Primary auditory cortex now projects to Wernicke's area, where meaning is connected to just the sound of the word. Okay, so Wernicke is putting meaning, allowing meaning to connect to the sound of particular words that come in. And then through this connection, number 3, the arcuate fasciculus, we bring the meaning of words to this preparatory area, Broca's area, this motor preparatory area, that then translates these words, the meaning of these words, into spoken language through connections to primary motor cortex. And in terms of the Geschwin model and the importance of connections among brain regions, we know that if you damage just the arcuate fasciculus, not even Wernicke's area and primary auditory cortex, which can happen rarely, what you see is patients that can't say words, patients that can't repeat spoken words, okay? So they can't repeat words that are spoken if you have an arcuate fasciculus lesion. Let's move to speaking a written word. So here you have visual input. So again, you should be familiar with all the pathways from the retina all the way to this area right here, which is primary visual cortex. Visual cortex, a written word, is processed by primary visual cortex and then projected to the angular gyrus. Angular gyrus is critical for translating visual words into their meaning, and that visual translation is then further processed in Wernicke's area, and then to the same pattern. Wernicke's area to Broca's area to speak it, and then finally to primary motor cortex to actually engage those motor muscles that are important for speaking the word. So what happens if you have angular gyrus lesions? Angular gyrus lesions prevent you from saying and repeated a visually seen word. So I could flash the word brain on the screen, and if you had an angular gyrus lesion, you would not be able to repeat that word. But if I said the word brain to you and asked you to repeat it, you would be able to repeat it. Why? Because all the pathways here are completely intact, whereas the angular gyrus is critical for that translation between visual word, the visual form, and spoken language. So this is a common model. You'll read about it in textbooks, you'll read about it in this textbook. But the problem is, in models like this, like many models like this, it's simply too simple. And the problem is that these areas are not nearly as specific for the things that Broca and Wernicke initially identified them for. For example, there are comprehension deficits in Broca's area. They may not be as severe or the same kind as the comprehension deficits in Wernicke's area, but they're still there. So it's not a pure motor area, it's motor combined with meaning. Similarly, Wernicke's area is not just comprehension, but it plays a role in certain kinds of motor output as well. So because this connectionist model was kind of defined by very unique functions of these brain areas, it's simply not a great example. Now, we don't have time to go into why it's not, but just have an appreciation that these models are a first stage in trying to understand, and we get more and more data in and try and understand why, how we must modify these models. Okay. So there are problems, they're not nearly as cleanly dissociated. That's what I just said. Okay, so now what I want to do in the last few minutes is talk about yet another kind of tool to understand higher cognitive functions. We've talked about lesions. We've talked about physiology. We've talked about just behavior in terms of specific tasks that might get at particular brain functions. So a lot of these are based on impairment of functions and learning about the brain based on impairment. What if there were people that had a super function, a super memory function, a super emotion or facial recognition, emotional facial recognition function, or a super language function? And it turns out there are cases of both. I don't have time to tell you about the super memory people, but some of us did anybody see the 60 Minutes show on people with super memories? And Mary Lou Henner, the actress from Taxi, who you may not remember at all, but she's an actress, has this amazing memory ability. Did anybody see that? Anyway, one example that I won't go into, but I'll just tell you for interest, you can Google it on YouTube, is that they've now discovered a subset of people that remember amazing amounts of things. And we never discovered them because they didn't think it was abnormal. But they can go back and I could ask you, okay, December 23rd, 2007, what did you watch on TV that night? And they'd be able to tell you what exactly they watched. And they'd be able to tell you whether there was a substitution, whether something was a rerun or not. And they have this amazing super memory. Well, it turns out that there's a syndrome that provides, that results in super language and musical abilities. It's called Williams syndrome. And in the film that I'm going to tell you about, one of the foremost world's expert on Williams syndrome is a woman named Ursula Bedlugi, who works at the Salk Institute in San Diego. That's where I did my PhD. And I'll never forget, one day I was walking up the walk to the Salk Institute to go to work one day. I was a graduate student there. And there was a little girl and her father walking in front of me. And she looked very much like this, kind of pixie-faced, which is very characteristics of Williams syndrome. And I walked past her, and she was very little. She looked like she was about 5 years old. And I walked past her, and I just walked past her. I wasn't going to interact or anything, as I was headed to work. And she said, "Hi, how are you?" And I said, "I'm fine." She said, "Do you work here?" And I said, "Yes, I do." She said, "What do you do?" And I said, I'm a graduate student. She said, oh, that's so interesting. I go and I visit, I work in a lab that studies language, and we're going there today. And she told me all about her day in a very, very charming way, and in a way that somebody, good conversationalist would do, and not the kind of banter you would expect from a little pixie-ish looking 5-year-old. She looked 5, I think she was probably older. And later I learned that this was one of the Williams syndrome patients that Ursula Balucci was studying. And so this syndrome is very unique. It is caused by the deletion of 28 genes from one of your two, you have 2 chromosome 7s. Each chromosome 7 typically has these 28 genes, and in Williams syndrome, one of those 28 genes in one of those copies of chromosome 7 is gone. So they have a lacking of one pair of genes, and they develop 2 amazing characteristics. One is amazing musical ability, rhythm ability, which usually requires great math skills, but they actually are terrible at math. They also develop very unusual language abilities with very unusual, large vocabularies. It's called the cocktail party syndrome because they are so great at a cocktail party. They can go and talk to anybody and be very interesting and very charming. And so you can ask, well, what do these genes encode? Genes encode proteins. So what protein is being encoded by these 28 genes? Well, we know what that is. It is a protein called elastin. And it is one of the things a protein does is causes the elasticity in your skin. And so that is what they think is causing the characteristic pixie-like look of these people. So Williams syndrome is not that common, and it's very difficult to study. But it's quite striking, and so I thought it would be great to be able to look at, we're going to look at 2 parts of this longer video. I recommend that you watch the whole thing. I'm just going to show you 2 parts. This is Oliver Sacks. You might remember him. He wrote The Man Who Mistook His Wife for a Hat. He's a famous neurologist that works at Columbia, and he's going to introduce this. I'm going to show you a little clip about all the Williams kids interacting with music. And then we're going to go to a part where they talk about language.
We arrived on Tuesday evening when there was a concert. And just as soon as it began, everyone there responded to the music. One realized this is amongst other things, a highly musical species.
Okay, so that gives you an idea of the musical capacity of people with Williams syndrome. And I'm just going to move to there she is. Okay, so this is Ursula Balucci. She's the one that works at Salk, who's an expert on Williams syndrome.
What we did at the very beginning, and I think it was an interesting way to go, was contrast age and IQ match Williams and Down syndrome children. Not because they're alike, but because they aren't. For example, on simple things like name all the animals you can in 60 seconds. we find Williams syndrome children almost at age level being remarkable, tending to do very unusual things. You get examples like ibex, saber-toothed tiger, chihuahua, albatross, and things of this sort, where the comparable Down's child goes in the other direction. It's very simple, just a few answers. What happens with the Williams syndrome children is that, on the whole, many of them are extremely good storytellers.
Mary and Joe were playing in the yard, and all of a sudden, this big, beautiful lamb came in the yard, and all of a sudden it talked to them, and told them different stories, and sang to them. And they didn't know where it came from. It just came out of clear blue sky one day. It just landed right there in their yard.
So that gives you a feeling for their language and why this is called kind of the cocktail parties. They can tell a fantastic story. And I just love the part of this interview where she talks about the kind of animals that they name. Ibex, saber-toothed tiger. These are like 7-year-olds giving ibex and saber-toothed tiger as typical, and just name all the animals that you can. Whereas a typical 7-year-old, let's say dog, cat, rat, if you're lucky. So very interesting syndrome. So what does this tell us? This tells us that there are multiple ways to study these higher level cognitive functions. It's not just all about lesions and what damage to the brain causes impairments. You can study people with extraordinary powers and learn something like that. This also introduces another kind of piece of the puzzle, and that is the molecular, the genetic underpinnings of language. So we think that whatever is encoded by these 28 genes missing in Williams syndrome is going to be a key to understanding how language is operating. We think that there are developmental changes that happen in these Williams syndrome kids, because sometimes they actually, at some part during their development, they have extraordinary abilities and they go away. While they have extraordinary music and language abilities, they cannot function in the real world. They can't tell time. They can't tell how long 5 seconds versus 5 hours is. They can't balance a checkbook. They can't understand how to do these basic things in the world. Yet he can do that rhythm thing that, I don't know, I think many of you and I in this classroom would have a hard time keeping up with the same syncopation that he's able to do very, very easily. So it's a long road between these 28 genes and understanding all the different ways the proteins from those genes interact during development and during adulthood to cause this extraordinary exuberance in language and music ability. But this is another direction. And in the last 5 minutes, we're going to have a little different exercise, I'd say in the last 15 minutes of class. I just want to remind you and summarize for you. Here we are. So we're almost done. Next lecture will be on the finals, so please come, Neurobiology of Love. But for this last third of the class in the higher cognitive functions, we've really been talking a lot about the same patterns of approaches as scientists that we've taken to understand higher cognitive functions. How have we learned about higher cognitive functions in the brain? We learned about them through lesions, through patient HM that had a surgical lesion that was a big mistake, and because of him, no more of these bilateral medial temporal lobe lesions were done because of the devastating effect it had on declarative memory. Downer identified the critical role of the amygdala in processing fearful information. Phineas Gage was our example for frontal lobe damage and brain damage, mainly through strokes, causing different kinds of aphasias. We also have learned from other patient populations, schizophrenia, and the higher cognitive functions that are affected in schizophrenia, and how that might happen and work in the normal brain. Williams syndrome as an exuberance of language and musical ability. we also will learn next time from animal model systems. So this is the same idea as we talked about in the bat. Bats are great at auditory processing, and we study the particularly well-developed auditory system of the bat to understand the principles of how the brain can process auditory information. In the same way, we study animal models that are really good at things like, well, I call it love, but people call it social bonding. So voles are one of the relatively small number of species that form lifelong pair bonds, male-female pair bonds. So they form this bond early on. We're going to talk about how, and even if the partner dies, you know, the poor other vole is alone for the rest of their life because they've formed this pair bond. But there are other vole species that are completely promiscuous. I mean, just kind of Saturday night going out to the discos, doesn't care, you know, one night stands all the time. So we could ask, what's different about the monogamous vole brain and the polygamous vole brain? And that's exactly what we're going to do next Wednesday, on Wednesday, not next Wednesday, but on Wednesday. So these are the kinds of approaches that we've used to try and understand higher brain functions. There's not a lot of them, but just appreciate that that is a valuable tool. So if you were scientists and faced with an alien species, you wanted to study the effects, the role of a particular brain area. Here are your tools, okay? And you should be able to take the examples that we provided you and to tell us the approaches that you would take to study such high-level function. Okay, so once we've identified a particular brain function, how do we study it in greater detail? We've talked about behavior, identifying tasks that are specific for particular brain functions. So differentiating, for example, between delayed non-matching to sample tasks, declarative memory tasks, and procedural tasks, those motor tracing tasks that HM was able to do perfectly well. He could learn and retain that information even though he didn't have declarative memories of that. And of course, the delayed response task that we talked about at the beginning of class. We talked about pathways involved. We can analyze pathways involved. We talked about the high road and the low road to understanding fear memory in the amygdala. We could identify not just pathways, but all areas involved in a particular brain function, like declarative memory. We talked about the studies that led to our understanding that the medial temporal lobe areas involved in declarative memory include the hippocampus, entorhinal cortex, perirhinal cortex, and parahippocampal cortex. These areas kind of focused around the medial temporal lobe and all surrounding the hippocampus. You can go deeper and understand the cellular underpinnings of this functions, plasticity, for example. You can study it in very simple systems like in aplesia, or you can study it in even more complex systems like LTP, studied in the hippocampus. You can study cell changes in things like schizophrenia, where we saw the messy organization of cells in the hippocampus. Do I know what that means? No, I don't. But that's just another tool to look at differential morphology in abnormal situations like schizophrenia. We can look at genetic differences that we talked about today, lack of genes on chromosome 7 in Williams syndrome, the lack of those 28 genes on chromosome 7. And this should have been higher, but we talked about you can record in brain areas as animals are performing that task. You think that brain area is involved in, and really understand what individual cells are sensitive to and how they're recording. So here I've just given you a bag full of tricks to understand higher cognitive functions in the brain. And it's variations of these tricks that we're still using today to understand. Really important point, we do not understand everything about the frontal lobe. Phineas Gage was not the end all and be all. There's still so much to understand about these areas. Same with the medial temporal lobe and declarative memory. But hopefully you'll get an appreciation, or you have an appreciation of the tools and how perhaps even more refined behavioral tasks or refined cellular understandings of this will take us to a deeper level. And that's exactly what neuroscientists are doing today. And so what I want to do in the last 15 minutes, what I should say, what we want to do in the next 15 minutes is we're going to have the TAs come down, and we're going to do a little review exercise for you in the form of 20 questions. So we've talked about lots of different syndromes and what different brain lesions can do, but what we want to do is give you an exercise where you actually have to figure out what exactly is wrong with the TAs. Okay, so they are going to be playing different roles, and you guys have 20 questions to figure out what their syndrome is. Who would like to, who wants to go first?
Yeah, I'll go first.
Okay, so Eric is going to go first. He has given me in a secret ballot the identity of his character that he's going to play today. And what I want to ask you to do is give 20 questions. Now, you're not allowed to say, you're not allowed, well, I'll tell you what you're allowed to do and not when you ask the question. But I want you to focus on the basic functions. And this is going to be from kind of the medial temporal lobe all the way to language inabilities, okay? So all of those syndromes that we covered from medial temporal lobe all the way to language. Okay, so and then we have a couple of other examples depending on how long it takes. Okay, who wants to start? Yes.
Do you remember what you had for breakfast this morning?
Breakfast this morning. Maybe have banana or oatmeal, or maybe it was a waffle. No, maybe it was pancakes. No, I think it was yogurt.
Question 2.
Where did you grow up?
Where did I grow up? I grew up in Michigan.
Yes.
Am I patient HM? My name's John.
Yes.
What was the first question we asked you?
First question, who asked me? I don't know.
Yes.
Do you have interrogate amnesia?
Do I? My memory is fine.
Yes.
What color are your shoes?
My shoes are brown.
Yes.
What did you do last year for your birthday?
Last year for my birthday, last year I turned 19, and it was a raucous party.
Yes.
What year is it?
What year is it? It is the year 2004.
I'm looking for a story to describe what John's situation. You could ask a question or you can try and tell a story. Okay.
John has a lesion in his campus. He did some damage and has anterior lesion as part of it.
Okay. What else does he have? What?
Partial retrograde amnesia.
Okay. Why do you say it's partial retrograde amnesia?
Because he can't remember last year, but he can remember like when he was born.
Perfect. Good. Okay. I'm going to say you, what?
How do we know you were born in Michigan? You grew up in Michigan, though?
I'm his doctor, so I can confirm that he was born in Michigan. That part was true. Okay, you guys win. You guys got the 20 questions. Great. Okay, you get your prize next week because I forgot to bring them. Sorry. All right. Next, patient. You could ask, so I am their physician. You could ask, what? Okay, oh, right. You could ask me the results of tests, if you like. Who wants to start?
What's your name?
Was there a question back there?
You occasionally hear voices.
Voices, no.
Yes.
I think you have aphasia. That was caused by probably because you can't understand what we're asking, you just can't communicate well, so you probably have Broca's aphasia.
Ding, 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 ding. Very good. Okay, 2 prizes. Very good. Thank you. Okay. It's going to be really hard. This next one's going to be really hard.
What's that supposed to mean?
We have 5 whole minutes to fill. Okay. Who would like to question our patient?
Yeah, that's right. You couldn't take me.
I scared them.
What's your mother's name?
Mary. Why? You have something to say about my mother?
Up there.
What do you want?
So you recently acquired a railroad spike in your frontal cortex.
Look at this face. What are you talking about? Beautiful.
When is the rat lab due?
Why are you worried about something like that right now?
Because it's relevant.
I don't even know what I'm supposed to be doing right now.
So the question was, when is the rat lab due? And she was a little belligerent. Next question. Yes. Okay.
Have you given your patient the Wisconsin card sorting test?
That's a very good question. Have I given the patient a Wisconsin card sorting test? And I have.
And how did their patient perform?
Poorly. I don't want to say it too loud.
I'm sorry, what?
Just relatively poorly. Yes.
Has your relationship with your friends or family kind of changed recently?
You know, they're all trollops. You just don't understand.
Okay. Yes.
So I think you have damage to the prefrontal cortex, and therefore you have less control over your emotions.
No, you have damage to the prefrontal.
Very good. Okay. So that is a 3D demo of these kinds of different syndromes. Of course, they could happen in multiple ways. You could have medial temporal lobe and frontal damage. Unfortunately, some people do. But just trying to use all the knowledge that you gained to understand the higher cognitive functions that we tend to take advantage or take for granted all the time. So, okay, I am around until 12:30. If you have any questions, please come see me. Next Wednesday is the last lecture. It's my favorite lecture of the whole year, so please don't miss it. The Neurobiology of Love. You'll learn all about sexy voles.